Hi, my name is Bijal Shah from the Moffitt Cancer Center. Thanks for inviting me to share with you guys the results of, or the preliminary results from the PBCAR0191 study. This is an allogeneic this is an allogeneic off-the-shelf CAR T-cell therapy that we use to treat patients with relapsed and refractory non-Hodgkin's lymphoma. This particular product is unique. It's generated using the ARCUS technology. And so I jokingly refer to this as a next generation CRISPR. Essentially, it is, though, a tool that allows you to very selectively, very specifically drop genetic payload exactly where you want it. And so in this case, we were able to do that to drop the CAR transgene directly into the T-cell receptor. So we can ablate the T-cell receptor and have the chimeric antigen receptor expressed on the T-cell all in one step. And so if you think about it now, we can minimize the risks, hopefully avoid them altogether, of graft versus host because we've taken out the T-cell receptor, and at the same time have that anti-CD19 effect that we want. And so what did we see, and why are we so highly encouraged by these data? One, what we observed is when we enhanced the lymphodepletion, and very specifically, all we did was increase the doses of fludarabine and cyclophosphamide. So, with very modest changes to lymphodepletion, we were able to generate really marked improvements in expansion. So now we can, really achieve high levels of CAR T cells in the blood, measurable by flow cytometry. And, this was coupled with high response rates. And so what we're seeing across heavily pretreated patients, diffuse large, mantle cell lymphoma, follicular lymphoma, you name it, we're seeing efficacy, to the tune of about a 78% response rate, complete remission rate, in these patients, with relapsed lymphoma, and that's translating into some durability. And so we do highlight, the early data we have again, and it is early, showing that, really upwards as far as 6 months in some cases, we're seeing responses. And then, finally, we highlighted just a case of a patient with, multiply relapsed follicular lymphoma, bulky disease, what we're seeing with this particular therapy. And it's one of the things that I love about this therapy is this concept of, delayed response. And because for me, as an oncologist, but also as a CAR T-cell, physician, what does that tell us about what's occurring within the tumor microenvironment, and potentially, is that something that's going to facilitate deeper and more prolonged remissions over time? So in this case, we gave the CAR T-cell therapy, and we saw by day 28 really a beautiful partial remission, but we could still see PET avid nodes, at this time point. Again, with further follow-up by day 60, we saw this completely, translate into a complete remission. And that's interesting. That to me is really interesting. And again, it's one of the things that I love to see on the trial. We saw it in a few other patients. And again, I'm hopeful that, that this is going to continue. I hinted at the fact that, I hope we don't see any graft versus host. We haven't seen any. In fact, I don't know that any meaningful graft versus host has been seen on any of the allogeneic CAR T-cell trials. So that, again, is also very encouraging news. We did also have a good sort of platform for safety in terms of low rates of CRS and neurotoxicity. And so, on the whole, really, really very pleased, really very encouraged. And, I'm really excited to see how are we going to move this forward, right, as we begin to identify the best T-cell. This is one of the strengths, and I've mentioned this before in the past, that we can now begin thinking about what is the optimal T-cell for infusion, because it's an allogeneic product, and so in the beginning, yes, we're stuck with monikers like central memory and naive T-cells, but there may be subsets even within those that facilitate deeper and more prolonged responses. I mean, if you have the right CAR T-cell, here's the amazing thing, it only takes one, right? I mean, you put it in the right, context where it can expand and you can generate remission. And so this is, some of the science behind the science, meaning the ARCUS technology, the ability to select out the best T cells, and the ability to generate these remissions by simply increasing the doses of fludarabine and cyclophosphamide. We don't have to worry about, again, making patients terribly sick in the process. So very encouraged, very excited, and eager to see where this program will end.